So hello everyone and welcome. Just wanted to make a note that your audio is muted, okay? So welcome to today's UVic Center for Biomedical Research Cafe Sci. You can hear my voice but not see me. I don't have great lighting in this room right now, so you'll just hear my voice for the intro. I'm Dr. Leanne Swain. I'm an associate professor in the Division of Medical Sciences at the University of Victoria. And I'll be acting as your host and question moderator tonight. So I will start by acknowledging with respect the Lekwungen peoples on whose traditional territory the University of Victoria stands, and the Songhees, Esquimalt, and Musanish peoples whose historical relationships with the land continue to this day. For those new to Cafe Scientifique, what we do is welcome scientific researchers like Dr. Karolina Valente, affiliated with UVic, to share their work with the community. Pre-COVID-19, as many of you remember, this was a nice sort of in-person event, but for the time being, we are doing this virtually and we're really happy to have it going. So we won't have any interruptions for Dr. Valente during the talk, so please note down your questions or post them to the chat as they come to you. So I have muted your microphones and they'll remain muted for the duration of the talk, which will run for 45 minutes. So after the talk, I'll be reading out your questions to our speaker. And that brings us to today's speaker introduction. So I'm super excited to have with us today Dr. Karolina Valente, who is a new assistant teaching professor in the biomedical engineering department at the University of Victoria. Congrats, Karolina. This is so exciting. Yeah, so Karolina has a bachelor and master's degree in chemical engineering from Lisbon, Portugal, and a PhD in mechanical engineering from UVic. During her PhD, Karolina studied better ways to deliver therapy to cancer cells, specifically within the breast cancer environment. Since her PhD convocation in August 2020, Karolina has received multiple postdoc offers from a number of prestigious institutes, but UVic was able to hold on to Karolina and recruit her as an assistant teaching professor, which aligned well with her passion for teaching and for this lovely city that we live in. So today, in her talk entitled Taking Advantage of Cancer, Exploring Drug Delivery Systems for Cancer Treatment, she will be talking about taking advantage of the cancer environment as a way to increase therapy efficacy. Yeah, and so also at the end, Dr. Valente will talk to us about her new company, Voxel Bioinformation or Bioinnovation, and I'm really excited to hear more about that. So welcome, Dr. Valente, and over to you.
Thank you, Dr. Swain. I really appreciate the kind introduction. So before I start diving into the topic of cancer, I thought it would be interesting to give you a little bit of background on myself. So as you can tell from the accent, I'm not from here. I was born in Brazil, more specifically Rio. And as naturally as it goes, I went to university also in Brazil. So I started my bachelor degree in chemical engineering, as Dr. Swain said, in Federal University of Rio de Janeiro. And it's the way that it works in Brazil is engineering is a 5-year program. And when I was around my 4th year, there was an opportunity to go and study abroad, to do some courses abroad. And then I got in and I got a position to go to Portugal and to do some courses in Portugal. So the plan originally was to go to Portugal, then come back to Brazil and graduate. So that's how I ended up in Técnico Lisboa. That is the best engineering institution in Portugal. So I managed my way to actually not just take some course, but to stay there. So I transferred my entire degree from Brazil to Portugal, graduated from my bachelor's, and started my master's degree also in Portugal, all in chemical engineering. And my master's thesis was also biomedical engineering related. What I did in my master's thesis was to design better membrane that I used in membrane blood oxygenators. So blood oxygenators are basically what the name says. It's those devices that oxygenate blood outside the body when the person is getting surgery. So that's what I did for my master's degree. And when I finished my master's, I started applying for PhD because I absolutely love the research side. So I applied all over Canada, but UVic got me, and just because it had exactly the areas that I wanted to work on, but also the nice weather, especially coming from Brazil, I do appreciate the hot weather that we have in here. So when I came to UVic, I went to the mechanical engineering department, and I was working under the supervision of two professors, Dr. Suleiman, that is from the mechanical engineering department, and Dr. Alex Brolo, that is from chemistry. So they're here. I got to work with amazing people from Dr. Suleiman and Dr. Brolo's group. And the picture there on top and in the corner, that's our last group picture that we took, and it was a post-COVID picture. As you can see, we're all working from home and doing our best. So yeah, so that's how I ended up here. So I graduated from my PhD last year in August 2020. And I will be talking a little bit about what I did on my PhD and what I'm doing right now and where I'm going research-wise. So going back to the topic, taking advantage of cancer, exploring drug delivery systems for cancer treatment. So when we look at cancer, especially on the focusing on Canada alone, we are talking about 1 in every 2 people getting cancer in Canada and 25% of people dying from cancer in Canada. So just in 2019 alone, was more than 80,000 deaths. So in terms of magnitude, I think it's easy to understand how heavy this topic is. So in 2019 alone, too, was more than 200,000 people were diagnosed with cancer just in Canada alone. If we look more on a health side perspective, there are different ways that we can treat cancer. There is a chemotherapy approach, there is the radiotherapy approach, and there is the hormonal therapy approach. So I'm going to cover a little bit of each one of them so you can kind of understand where this conversation is going to go. So the chemotherapy, those are drugs that are injected in your blood vessels, and they go throughout the entire body. And what that drug is targeted to do is to stop the growth of cells that are growing uncontrollably. So cells that are growing quite a lot and they can't stop growing. Because that's one of the main characteristics of cancer, is the mutation on the genes that they have that can't stop proliferating. So, and that's why when people receive chemotherapy treatment, they also lose their hair, because hair cells are also cells that keep growing very, very fast. So the chemotherapy is affecting the entire body, not just the cancer cells. When we go to radiotherapy, the second one here, it's a bit more target with the X-ray beams. The problem with radiotherapy is it does work very well when you have a superficial tumor or a tumor that is located in a certain area. It does not work well when it's all over the body. That's because the penetration of those beams, they are inversely proportional in terms of efficacy, inversely proportional to that. So the more you go into the tissue, the less that therapy will have an effect in terms of the cancer. And hormonal therapy is a different type of treatment. It's targeted more to cells, to cancer cells that have hormonal receptors for estrogen, progesterone receptors. So the cancer cell, the cancer therapy goes to the cancer cells and kind of fits in that receptor. So I started looking into all of this and thinking, well, there is these different types of treatments. And also, if we look into cancer, for example, 2 people with the same cancer, they will react a bit differently. And that's the main focus on the literature out there. What is the difference? But from a mechanical perspective, a mechanical engineering perspective, I started looking into what do we have in common? What do all cancers or most cancers have in common in terms of what is happening in that healthy tissue? So that's how I did my PhD. I started looking, well, when we have cancer, what is actually happening in that tissue? So imagine you have a healthy tissue and you start having cancer cells growing within the healthy tissue. What the cancer cells are going to do, they're going to start squeezing the healthy tissue. And that entire tissue now that has cancer becomes pressurized. So you see the effect of high interstitial pressure in that area right now. Because now you have a high interstitial pressure, you also have a lot of stiffening happening on this area. And the stiffening is coming because of this pressure, in addition to accumulation of the extracellular matrix. So when you look at a tissue itself, a tissue is not just cells. A tissue is composed by cells plus a scaffold. So a scaffold is basically where cells are sitting in order to give the shape for the tissue. So you start seeing that scaffold becomes stiffer and stiffer in addition to the high interstitial pressure. The third option that occurs, the third thing that occurs is the hypoxic regions. The hypoxic regions, those are regions within the tissue that there is a lack of oxygenation, which means that the cells at that point are lacking oxygen. And that's a bit dangerous. And let's take a look why. So this is the image of how the drawing of how a tissue looks like. So you have the blue cells in here that are the cancer cells, they are growing within the tissue. You have these yellow lines that are showing the scaffold. You have the yellow cells that are the healthy cells. But now the tumor is growing there and the tumor needs to be fed. The same way that for our healthy cells, the capillary vessels are pressurized and nutrients migrate from the capillary vessels into the healthy cells, nutrients, oxygen, and so on, by this pressurized environment, by this pressure-driven movement. When the tumor is growing there, the tumor also needs to be fed. So what these tumor cells do is they start sending signals to the blood vessels and telling the blood vessels, well, now you have to feed me. So what the blood vessels start doing is they start sprouting and going all the way to feed those cancer cells. So now it becomes a bit dangerous because the cancer now has the nutrient and it starts growing even more. So if you look at these images here, you can see this is a normal vasculature for a healthy tissue, while this is the vasculature for a tumor environment. You can see it's very, very disorganized. So this is what is happening when you have cancer in a healthy tissue. So if we look at breast right here, before cancer, we have a stiffness that is an elastic modulus, that are mechanical properties of the tissue. That is around 400 Pascal in here. But once there is cancer in that environment, that increases the stiffness quite a lot by changing the environment. So I started thinking a little bit more about that. And now, because before in a healthy tissue, you have capillary vessels that are pressurized and things are moving from the capillary vessels into the healthy tissue by a pressure difference. Now, because the cancer is growing there and is pressurizing the tissue, you don't have this pressure difference anymore. This pressure, the pressure in the cancer and in this tissue now that contains cancer is equivalent to the pressure of the capillary vessel. So things are going to move from the capillary vessels, and by things I mean not just nutrients and oxygen, but also the chemotherapy drugs and whatever drug you are injecting there. They're going to move now by diffusion. And diffusion is very challenging, but is what I studied the most in my PhD. And just to give you an example of what I mean by diffusion is the following. The difference between convection and diffusion can be easily explained by if you imagine having a glass of water. So you have a glass of water, and then you pipette or you drop in there a few drops of food coloring. If you use a spoon to mix, what you're doing is that you're applying an external force and you're forcing the molecules from this food dye to mix with the molecules of water until you have a homogeneous concentration in the cup imagining being the food dye blue, then now you have a homogeneous concentration in the cup. But diffusion would be more you dropping those food dyes and not doing anything, and letting the dye molecules by themselves to go around the water cup, the cup of water, and by themselves to create a homogeneous concentration. Because diffusion is very slow, but the molecules will go from a high concentration gradient all the way to a low concentration gradient. So thinking about that in the concept of cancer, I wanted to understand what is the impact of diffusion in the delivery of drugs in the cancer tissue. Because now, again, I don't have the pressure gradient anymore. And to understand that, I decided to use different tools. One of the tools that I used was micro-fluidic devices. So they are exactly like you're looking here. So they are small devices that are smaller than your credit card, and they contain very tiny channels, as thin as your hair. So inside of these channels, you have a very high spatial and temporal control of the fluid that is passing there. And you also are using a very small reagent volume, so it's very cost effective. And the idea with the microfluidic device is now the dimension and the 3D environment can be used to mimic some architecture, some physiology components of our body. So I thought, okay, I'm going to use microfluidic devices to then mimic the cancer environment and mimic my diffusion in an in vitro system. And by in vitro, I mean in glassware in the lab. But although I can have a very high control of diffusion in these systems, I need something to mimic my tumor. So I can have cancer cells, but as I said, a tissue itself is not just composed by cells, it's also composed of an extracellular matrix that is the scaffold for that tissue. So to create that scaffold, and even though you may not have heard the name, you probably have seen this before. I used hydrogels. So hydrogels are materials that you find in contact lenses and all these, also these wet band-aids and so on. So they are polymeric materials that are very biocompatible because they are hydrophilic, which means that they like water. So they swell a lot in water and they resemble a real natural tissue. When the cells are in contact with this environment or encapsulated in this hydrogel, the cells quite like that. Okay, so I have my number 2. I have how to mimic diffusion. I have how to create my tissue in 3D. My third point was, well, studying chemotherapy drugs, it is interesting, but I wanted to take a step further and go a little bit more towards what my PhD supervisor, Dr. Brolo, does, that is gold nanoparticles. And you heard exactly what I said. It's right. It's gold nanoparticles. So there are particles made out of gold. And by nano, I mean really, really tiny, 10 to the power 9, minus 9 meter. So going back to the radiation therapy, I have an equation here that shows you radiation enhancement. And this radiation enhancement is dependable on the energy that you are applying, but also on the atomic number. That means the number of protons that you have in that environment. So if you are calculating radiation enhancement for a red tissue, your atomic number is around 7. So that's a pretty much constant. So you can easily calculate how much enhancement you're going to have in that area. But if we use metallic particles, because of their very high atomic number, what will happen is, Ian, I'm sorry, can you still hear me? I heard a little beep.
Yes, I heard a little tone as well. I was checking if it was the chat, but I'm not really sure where it's coming from.
Okay.
Yeah, I don't think it's, yeah. Sorry, Karolina, I just want to add, I don't think it's, I turned off all the notifications, so I'm not sure where that came from.
No problem. I'm just checking if I'm still here. Thank you. Okay, so I'm going back to the gold nanoparticles that are these very, very tiny particles made out of gold and comparing the atomic number of a tissue that is around 7 to the atomic number of a gold nanoparticle that has an atomic number of 79, you can see how much enhancement you have if you have gold nanoparticles in the environment where the cancer is. So if we find a way to put those particles in there, when you heat with the radiation, that can have a much higher effect in killing the cancer cells by enhancing the radiation power. So these are the third tool that I use to analyze some parameters in my PhD. So the project was fairly simple. Let's create a tumor microenvironment on a chip that is inside of a micro-fluidic device. And I focus on breast cancer. So the chip was composed of 3 main channels. You have the central channel here that is labeled in blue. That is mimicking the extracellular matrix. That is mimicking that scaffold for the tissue and the cells and so on. And you have 2 lateral channels that are labeled in red that are mimicking the blood vessels. And that's how I'm going to be tracking the diffusion. What I was injecting in these blood vessels was gold nanoparticles, and there were 10 nanometers gold nanoparticles, really, really tiny, and a dye called fluorescein. And I focus on fluorescein because fluorescein has the same size of most chemotherapy drugs, so the diffusion is very comparable to chemotherapy drugs. But then I took a step further, in which in the blue region here, I mimic not just a cancerous tissue that was a sieve, but I also mimic a soft tissue that was comparable to a healthy tissue. So I wanted to quantitatively know how much the effect of stiffness and pressure was having on delivering the therapies to that tissue. So to create the scaffold, I use a combination of hydrogels made out of collagen, that is a natural polymer that you have in your body, that is a natural component of your extracellular matrix, and I synthesize a material called gelatin methacrylol. That's a very biocompatible material that is made out of gelatin. And by looking at the literature, I noticed that the breast tissue, when it's healthy, is between 150 and 400 Pascal in terms of elastic modulus. Going to a breast cancer, it increases quite a lot. As you can see, it goes all the way to 8 kilopascal. So by mixing these 2 components, I came up with a concentration for a healthy tissue that was around 200 Pascal, so within the range that we wanted, and around 6 kilopascal within the range that we also wanted. What is interesting about hydrogels is, if you think about regular tests to measure this elasticity of the tissue, for regular polymers, you basically do the tensile test, in which you have your sample here, and you kind of apply tensile stress this way, and you measure the deformation on your sample. The problem with hydrogels is they're very soft. They resemble human tissue, so you can't do this type of measurements. So by using a modified Hertz model, all these crazy equations here, we created a way to analyze these hydrogels at UVic. And so the way that I did was I created a pellet of this hydrogel, so a small cylindrical structure, and I labeled in green. And I applied in the surface of this structure, I applied some red beads. They're just sitting at the surface. And I came very carefully under the microscope with a spherical indenter and literally a ball bearing from a defined mass and defined composition. And I tracked the deformation that was created at the surface by having these profile images here. So by knowing how much deformation was created, I could know exactly how much was my elastic modulus. So now I have these 2 matrices, these 2 composites that can mimic healthy and cancerous tissue. But do my cells like them? Can I put that matrix material in contact with the cells, and will they enjoy? So by looking at what the cells look like in these constructs, Green means happy, means live. Red means bad, so we have a little bit in here. And the blue is labeling the nuclei. So going a bit further, I have a better image here. You can see, oh, sorry, going back. Okay, this image here in the middle is showing how the cancer cells display in 2D. And by 2D, I mean just a petri dish hard surface. But once I put these cancer cells inside of my polymeric material, you see they form these very, very massive spheroids, mimicking exactly like cancer looks like in the body. So the cells were happy, and now I had all the tools that I needed to measure the diffusion and measure the impact of the tumor microenvironment on my chip. So by tracking the diffusion from the lateral channels all the way to the central channel, and by doing some calculations, I was able to see that going from a healthy tissue into a cancerous tissue, you are decreasing the diffusion by 70%. And that's how much of an impact the environment of the tissue has. In addition to that, we also saw that when mimicking the cancerous environment, it was very similar for the values that we got for the gold nanoparticles compared to a value that was displayed in the literature for immunoglobulin. That is a protein that is more or less the same size as our gold nanoparticle. So they displayed the same values for diffusion coefficient. The difference is the value in the literature was done in vivo in a mice. So, which means that our simple, well, relatively simple device could mimic in vivo conditions, which was very exciting. And after that, we had to make sure that the gold was actually going inside the cells. So we cross-sectioned the tissue, checked it inside the cells, and fair enough, the gold nanoparticles are going there. The problem with these gold nanoparticles is, okay, now these two to enhance radiation. The problem is, as I said, these particles are very, very small. They're 10 nanometers. So when you inject these particles into someone, they go all over the body, and the kidney filters most of these particles. So you basically pee them out. But if these particles were larger, what will happen is your body also capture those large particles, especially your liver, capture those large particles and also dispose them. So I started thinking, is there any way that I can create a more target delivery here for the cancer environment? So if we go back and remember, when the cancer cells are growing, they start sending signals to the blood vessels and telling the blood vessels to kind of feed them. The growth of that blood vessel is very, very fast, which means that you get blood vessels in that cancer area that are what we call leaky. So you have these gaps between the walls of the blood vessels, and those gaps now can be used to deliver stuff directly to the cancer cells. So diffusion takes time. It takes even more time if something is big, because diffusion is inversely proportional to size. So if you have small particles, they will diffuse well, but they will also go all over the body. If you have large particles, they will take time, but they may accumulate in the cancer area. So my point was, what if I create a particle that is labeled here in blue, and that particle contains my gold nanoparticles inside? Basically have a capsule that is the size that I want. Can I keep going? Yeah, okay. A capsule that is the size that I want, that to deliver and to pass through these gaps directly, but then inside contains my gold nanoparticles. And all of this is again based on the phenomenon that we absolutely love, that is the diffusion. So the idea was relatively simple again. Let's have a particle that will then deliver these small particles. But then it came the other question, well, how do I do this delivery? How can that be done? So going back to the tumor environment and thinking about high interstitial pressure in there, because the pressure is so high, clearing up the toxins is also very difficult, which means that now in that cancer area in the tissue, you have an acidic environment because you have a buildup of toxins. So I decided to create a polymer that would then respond to this environment, and it would then swell in acidic pH, releasing those gold nanoparticles. The interesting part of this project is I created the particles inside of a micro-fluidic device, as you can see here. So those are the images of the particles. They're around 100 nanometers. And without the gold and containing the gold, there are these tiny black spots. So we are very excited about this part. That was my last project on the PhD, and Dr. Brolo now has students working on this project to see what is the potential that we have there. So I kind of stopped at this area and decided to look more towards the tissue engineering and 3D bioprinting. My whole point was when I was creating this breast cancer tissue, I found the proper polymer to create this scaffold and to put the cells there, and the cells liked. They create those beautiful spheroids, but I couldn't find a tool to create the proper shape of the tissue. 3D bioprinting does exactly what the name says, is bioprinted tissue in 3D. So basically it's creating this 2D morphology layer by layer, piling them up until you have 3D tissue. But the techniques available in terms of resolution for 3D bioprinting were not good enough to mimic the main physiology components that we have in the blood, in the body. In addition to that, if we think about the whole drug development process, these tools could be used for that and could be enhancing the process of drug development. So if you look in terms of the drug development, you have something that starts with lab research and then moves to animal testing and then moves to people. And then finally, you hopefully have a drug that is approved. This process can take up to 15 years and can cost more than $2 billion. And the important message here is that a 90% failure rate. And that's as simple as because we are testing cells in a monolayer and then comparing those results to animal testing and then trying to correlate animal testing to us. And the correlation between these tests is actually less than 8%. So we should incorporate 3D bioprinting and tissue engineering into this process in order to enhance the translation of these results, to have a higher number, to decrease this long process. The problem with the 3D bioprinting techniques available is they display an average of 100 micrometers resolution-wise, and that's the thickness of a hair, while some of our blood vessels are actually 20 times smaller. To solve this problem, that's when I created Voxel Bioinnovation right after my PhD. So Voxel Bioinnovation is a new biotech startup located in Victoria, BC, and we are developing Canada's first high-resolution 3D bioprinter. And the main point in here is truly biomimetic. Our mission is to create tissues that resemble the human body. So if you take a look in terms of magnitude and resolution that we can go, this is an image of a petri dish, and I'm holding an Eiffel Tower there, and it's really tiny, and that's how it looks like amplified. So that tool combined to a vasculature network software that we are developing is going to be able to produce these 3D in vitro models that can now be tested towards the cancer development in order to study diffusion and convection in a more meaningful way. And of course, I had this idea, but I didn't do this alone. I have an amazing team supporting me, and they are all here, all these beautiful people, including my PhD supervisor, Dr. Alex Brolo. So the main point with Voxel, it is to become the 3D bioprinter for industry and academia. And when we compare that to the drug development process, and going back to those 15 years and more than $2 billion, what we are trying to do is to narrow this down, is to shorten the time, is to increase the translation, is to take everything that I learned from my PhD and apply now towards this company in order to create this meaningful tool that cannot just be used by academic institutions, but also to be used towards the drug development field and towards cancer therapy. So as you saw from my presentation, I approach the life sciences field a bit different than everyone else. So instead of looking at what we have differently, how is the cancer behaving differently in different people, I look at what we have in common, our similarities. And that's what I truly believe to be the proper approach towards a better world. Thank you very much.
Thank you so much, Dr. Valente. Excellent talk. So just a reminder, and I think I can share my video now. I've worked on the lighting in my room as we were chatting, as you were talking. And so I would just welcome people to post their questions to the chat, and I will read these off. I'm already getting some messages of great job, Karolina, so that's awesome. So maybe I'll just start off with a question to get things rolling, to get the ball rolling for people. Okay, so I was just wondering, with some of your results where you found the gold particles coming into the cell, what compartment did you find them in, and how do you think that what do you think the implications of that are? So where did they go into the cell, and does that impact how they could be used as treatments?
Yeah, absolutely. So when I tried to kind of summarize a little bit, but when I was using these particles, I tried different surface properties, so different surface charges to see if they would go to different locations in the cell. But what I found is these 10-nanometer particles, they normally go to vesicles, which is good because that's how the cell brings the stuff inside. But they don't go to any specific location in terms of, for example, the nuclei or the mitochondria or so on. That would make it maybe a bit more interesting. But the good part about them going to vesicles is, as you can see here in this image, they do go in a large quantity. So we were able, by using ICP-MS, so a mass spec, to kind of quantify the particles. And what we actually saw is positively charged particles. They are uptaken the most. So that was an interesting result, because if you think about using these nanoparticles, you need to know the impact of the surface charge on the uptake. So when we started going towards the nanogels with gold inside towards this particle, we had already narrowed down which type of gold nanoparticle we were going to include in here. That was the positively charged particles. Does that kind of answer your question?
Yeah, definitely. Yeah. Oh, great. We have a question in the chat. So this is from Jaden Foley. Once you're able to create fully biomimetic tissue using the 3D printer, are you planning on returning to the work with gold nanoparticles, but now using the new tissue constructs?
Absolutely. So that's a nice question, because what I learned from my PhD, I'm now trying to apply towards the company and towards this too, is by combining the diffusion and by combining the gold nanoparticles all the way to now to a tissue that can actually resemble the human body in a meaningful way. So that's the ultimate goal, is to be able to recreate cancer tissues in a more physiological manner than we were able to do before, and definitely go towards testing gold nanoparticles for radiotherapy, and also different types of drug delivery system that we also approaching Voxel. Thank you, Jaden, for your question.
Thanks, Jaden. So while we wait for another question from the chat, I had another question for you. So just in terms of like the choice of gold over other metals and thinking about some of the similar metals that we have endogenously in our bodies, are there any sort of issues or concerns with like homeostasis of those other metals like copper and zinc? Can gold, the presence of gold, sort of interfere with endogenous processes, or do you have any insights or comments on that?
I don't have as much of a knowledge of the interaction. What I do know is the choice of gold is due to the high atomic number. And what is interesting about that is there are many studies about cytotoxic of drugs that have, or like gold nanoparticles inside the body, and assessing the cytotoxicity, we have been seeing that gold is not as bad. So now we have groups that are already doing the clinical trial towards using gold nanoparticles for neck kind of tumors. So enhance the radiotherapy and also does not have a high effect in terms of killing cells or damaging the body. So it is, I don't think we can say that it's biocompatible fully, but it's definitely better than we originally thought when dealing with gold.
That makes sense. Great answer. So we have another question from the chat. So this is from Ariadne. Great job. What was the most challenging aspect of your studies?
I think having all these ideas from my mind that came and then continually being creative was definitely something that was challenging during the PhD. But what I think was the most challenging was definitely the diffusion experiment. So tracking the diffusion inside of a micro-fluidic device is very time consuming. The diffusion takes a long, long time. And feeding all these equations by using different Fick's law of diffusion, the second law of diffusion, for the different models that we have in here that consider having pressure, not having pressure, having equal pressure, and so on, was definitely one of the most challenging parts. But once I finished this project, that was my first project in my PhD, and I was able to see that quantitatively the meaningful information that we were getting comparing a healthy tissue with a cancerous tissue, and comparing with the rest of the literature, seeing that our numbers were actually mimicking what was happening in small animals was definitely very rewarding, but it was challenging. Yeah.
Nice. Okay, so while we wait for, we have another question. So a question from Laura. How long are the tumor constructs stable? Do they eventually break down and become unusable?
Yeah, so the thing with hydrogels, I'm just going to go back there, Laura. The thing with hydrogels is they do degrade over time. So if you think about applying hydrogels for tissue engineering, you want to have this hydrogel with the cells, supporting the cells. And if you're implanting that in the body, you do want that hydrogel to eventually degrade, and then the cells kind of integrate in the tissue, right? So you want that integration. So the hydrogels, they have this one of the main functions for them is to degrade. So for the tissues that I was dealing with, the composites in here, I let the cells grow within that tissue, within the consorts, in order to create these massive spheroids for 7 days. And then we did the gold experiments and all the diffusion experiments for 2 days. We tracked it for 48 hours. So it was 9 days. And what I analyzed is the constructs started breaking down after 2 weeks. So they had a lifetime of 14 days with the same mechanical properties. After 14 days, the mechanical properties started varying because they started degrading. So yes, 2 weeks.
Very cool. So I had just another, it looks like Laura had a follow-up question. Was the construct breakdown frustrating to deal with?
Was the construct what?
Was the construct breakdown frustrating to deal with?
Well, from the tissue engineering perspective, every time things are falling apart, it's definitely very, very frustrating to deal with. I was lucky enough to be working with cancer cells, because what we learned about cancer cells is they grow very fast. And they're also tough cells, so they kind of survive most of the time. So I didn't have to wait, for example, when we are doing with experiments with neurons or stem cells, I have to wait weeks for those cells to grow. I didn't have to deal with that much. So it was frustrating a little bit, but not as much. It was more challenging when we went towards when I went towards detecting the gold nanoparticles inside of those tissues, that you have to deal with all the sectioning of the tissue and so on to do microscopy. So that was a bit more challenging in that aspect. But the breaking down, I can see from having someone and having friends in the neuroscience department and so on, it may be more challenging with other cell lines than it was for me.
Nice, for sure. That was a great answer. As a neuroscientist, I can confirm. Yeah. So we have another question from Ariadne. If you had unlimited funding, what would you like to investigate?
If I had unlimited funding? Yes. Okay, definitely all these results, especially for my PhD, but definitely all these results in vivo. So everything that was in terms of diffusion for the gold nanoparticles and for the dye, using real chemotherapy drugs and investigating that in vivo is definitely one of the main things that I wanted to do. And then after that, take a step further, that is actually applying radiotherapy to that tumor. So the main point right now is having someone working in these nanogels with the gold inside and kind of tracking the delivery. That's where I'm hoping this project is going to go towards now in vivo studies. And then combining what Voxel is trying to do, which what I tried to do during my PhD, that is compare now the in vivo studies that we will have for these particles with what we can do in terms of the tissue and voxel, and kind of comparing the two results to have a more meaningful information of the impact of 3D bioprinting in the drug development process and the liver of therapy.
That totally makes sense. Yeah, thanks for the questions, Ariadne. So we have another question now from Ilse Stevenson. So a Sunnybrook group is working on using ultrasound to burst therapeutic nanobubbles within brain tumors, but stiff tissues will still affect diffusion of these agents within the tumor. Does your work give any clues to how to change diffusion characteristics within tumors?
I did not focus on that. There are some people focusing on how to change and how to target the extracellular matrix instead of targeting the cells itself. And by targeting, I mean by changing the mechanical properties, by inducing some degradation of the extracellular matrix that is stiffer at that area. We did not do that, but it's definitely something that would be very interesting to know a bit more.
Yeah, perhaps maybe a combination of gold nanoparticles and extracellular matrix disruption could actually increase infiltration of the gold nanoparticles right in.
Yeah, thank you for your question. That was very interesting.
Yeah, it's a very, very good question. So do we have any other questions? Get your questions into the chat. I'll ask another question as we wait for another one to come up.
Absolutely.
Yeah, so this is maybe for the students either in the audience tonight or who might watch this later on YouTube. So as a recent PhD grad, can you speak to how the skill development that you had during your PhD facilitated your transition into developing this biotech company, in addition to your work as an assistant teaching professor? So how did your skill development in your PhD facilitate taking on these roles?
What was interesting about the PhD was I wasn't expecting to have the amount of freedom that I had when working with Dr. Su and Dr. Bro. And that was actually worked so well for me because I was able to develop my own projects, to have my own ideas. And by having this creativity gear always pumping on my head, that's how I came up with Voxel Bioinnovation. But not just that, not just the creativity side, but also being able to see exactly what is going on this cancer field, on the tissue engineering field, and kind of being able to pinpoint what is lacking, what can we do better, and why are we not doing better. And especially talking about the drug development process, why are we still taking 15 years and more than $2 billion? How can we change that? So the skills from the PhD was definitely learning a lot from the literature, learning a lot how to deal with frustration, and instead of letting that hold me back, it's making me see another way to approach the problem. So every time an experiment that did not work, it kind of made me sad, of course, but it did not hold me back. It was just one more approach that it didn't work. So last one thing that I need to worry about, and now is trying something else. So having these very strong background in the literature and what is happening, and having this autonomy to kind of trying everything that I wanted to try and learning all these techniques, it definitely made me the person that I am today. I always joke about saying that everything that I learned during my PhD. And most of my background did come from there. So it was a very, very important moment of my life, very valuable moment of my life of learning how to deal with frustration, learning how to multitask, having a deeper understanding of the literature, and being able to communicate my ideas in a way that is not just understandable to the people in my field, but also to the general public. So, yeah.
Nice. And that's really evident, I would say, in your presentation today, that you have a lot of skill in distilling complex information into ways that are universally understandable. So, and your images are very nice, and really nicely put together presentation.
Thank you.
Yeah. So I think we have maybe another question that's coming in the chat. So yeah, I just welcome an additional few questions here in our last few minutes that we have. And maybe I can ask Karolina another question as we're waiting for that.
Yeah.
So do you have any advice to people who are just embarking on their graduate degrees in terms of, like, you mentioned that, sometimes things don't work. I mean, oftentimes in the lab things don't work and you have to be creative and find a new way. So do you have any advice for new students that are embarking on that journey?
I have 3 advice. The first one is persistence and hard working. That's the only way that I see of getting things done in a meaningful way. It's being persistent and trying again and trying again. Which leads me to my second advice, that is, when an experiment fails, it does not mean that you fail. And start not taking this too personally. That's a very important lesson in here that I learned during my PhD. Is when one experiment fails, that's it. The experiment failed, and then you move on from that. You redesign the experiment. And the third one, if my supervisor is here watching, he may not like it. But sometimes when people say, yeah, that may not be a good idea, but if you think it is, just try. Just take a little bit of time and effort to try, and to try to show to people why that's a good idea. So using facts in the science approach is definitely it's going to work to your benefit.
Absolutely. So to Rilla and Rich, who have their hand up, if you could post your message in the chat, that would be great, and I'll pass that on.
Sorry?
I guess there's a group, one of the participants has their hand up.
Okay,
but we would just ask, if, I think it's Rilla and Rich. So if we could have them just type it in the chat. Looks like they maybe put their hand down. I don't see it coming up in the chat. So, well, that was like amazing, amazing advice, I think, to new graduate students. And Oh, there's just a mistake. Okay, no worries at all. Yes, so very appreciated, Dr. Valente. And if you have any other final comments that you want to make, it would be great to hear them. Happy to hear. We have a few minutes left, so if there's anything else you wanted to add, anything you thought of at the last minute here, go ahead. Advice, anything.
I was just gonna say, by teaching the biomedical engineering students right now has been quite rewarding. I have been teaching them since I was a PhD student, so I did both jobs for a while. The approach that I take with my students is the same approach that I take with everything. It's just try and then try again and work hard, and that it will pay off. There is no other way around it. It will pay off. Don't be afraid of asking, and also don't be afraid of asking for help. That's an important lesson here, because even though most of these projects that I showed you and most of the results that I showed you, I did it alone. I didn't do 100% of it alone. I did most of the experiments alone, but I had help and I had support, and having this support is very, very important. And having this amazing group that I had with Dr. Verolo's lab supporting me throughout this journey was very, very important. So don't be afraid of asking for help if you need help, or at least asking for guidance to put you in the proper direction. And that's something that I tell my students all the time. There is no such a thing as a stupid question. Questions are just questions, and I'm here to clarify all of them. So I hope you enjoyed. I hope you guys check it out, Voxel, even more. And we do have been hiring co-op students too, so check it out, because we also may have more positions for later on if we have some students in here watching this talk.
That's amazing. Thank you so much, Dr. Valente. That was a fantastic presentation and tons of excellent, outstanding advice for grad students out there and budding scientists. Yeah, that's fantastic. And just another comment to relay from the chat from Kim Polglase. Thank you, Carol, Lee, and Ian for the great presentation. So yeah. Congrats on amazing presentation and amazing body of work and your PhD and your new positions as a professor as well as a CEO. You're just amazing, and yeah, keep up the great work. Very inspiring.
Thank you so much. Thank you, Dr. Swain. I really, really appreciate all the kind words, and thank you for all of you for coming here and watching me. I really, really appreciate it.
Absolutely, I echo that. So I think we'll thank you so much, to the group. Lots of thank yous coming into the chat, and thanks everyone for attending. The cafe is only, its component parts, and so the participants and the speakers make it what it is. We just really appreciate everyone's participation, and even though we've got these restrictions and sort of barriers, we're breaking through them in order to keep the science going, keep the communication going, and well done on a talk tonight. So great to hear all your amazing work.
Thank you.
I'll stop the recording now.